This video is going to show you how to do the lab that I've assigned for today. So if you read the instructions on the left-hand side, it tells you to click on that TV monitor to watch the video about the cell cycle, so you should do that. And then you're going to click on the information button to learn about cancer. And then you click on the microscope to begin actually doing the lab. So you will click on this microscope, and this is the screen that comes up. So what you're looking at here is you're looking at normal lung tissue, and you can see that because it says normal lung right there by the microscope. And so what you're going to do here is you're going to identify these cells, decide what phase they're in, and then click and drag the right tag to the cell. So I'm going to click and drag the ones that are interphase. So that one's interphase. Here's another one that's interphase. There's one that's interphase. And there's another one that's interphase. And then this one is prophase, so that you can see the chromosomes are in there, but they're not really in much of an order. So that's prophase. Then once you have all of those there, you check it. And it's going to label every cell. So you don't have to figure out every one of them. It labels them for you. But at this point, you do have to count all of the cells. So you count up all of them that are in interphase, and then count all of the ones in the other. So then once you have all of those counted and you have entered those numbers into the table in the normal lung row, then you need to change to a different tissue type. So the second tissue type on there is normal stomach tissue. So to get to that, you click on the box of tissue slides. And then you choose normal stomach, and then it will automatically bring up the slide for normal stomach. And you're going to do the same thing again. You're going to put the labels in for the cells. You're going to identify the phase that they're in. Then you'll check it, and you will write down all of the ones in interphase, and then each of the 4 stages of mitosis. Then you do that with the normal ovary. And so then you'll have that part of the chart filled in. And then the next part is to do the cancerous tissue. So you'll go here and you'll choose cancerous lung tissue. Again, you're doing the same thing, identifying those phases, checking it, and then counting up how many are in interphase, and then the 4 stages of mitosis. So once you get that table, actually those 2 tables all filled out, so you have your numbers for the regular lung tissue, the regular stomach tissue, so on, and you also have your numbers for the cancerous tissues, then you're going to do the back side of this paper. And what it wants you to do is it wants you to come up with an average percent cells at rest and a mitotic index. So the average percent cells at rest is just the percentage of cells that are in interphase. The mitotic index is the percentage of the cells that are actually going through mitosis, so all 4 of those stages. So what you'll do is looking at your data, and I'm just going to kind of make some data up. So let's say that you had 18 cells in interphase, 1 in prophase, 0 metaphase, 0 anaphase, and 1 in telophase. Then what you're going to do in order to find the percent that are at rest is you would add all these up, so 18, 19, 20 cells total, and you're going to take 18 divided by 20 to get the percent that are at rest. Then in order to find the mitotic index, you will take the cells that are going through mitosis. So you add 1, 0, 0, and 1. You have 2 cells going through mitosis. You're going to divide 2 by 20, and that's going to give you your mitotic index. So that tells you what percentage are actually going through mitosis. Okay? So that's how you will get the numbers for this part, these data tables. And then when you answer the questions, you'll basically be comparing normal tissues to the cancerous tissues to see if there's a difference between the average percent of cells at rest and the average percent of cells that are going through mitosis at any given time. Okay? And that's pretty much it. Good luck.